Okay, so now I think we have like 25 minutes or so for general discussion for all the talks. And let me first go back to a couple of questions which we had before. And one of those was from, excuse me, I forgot the name already. Peggy, I'm not sure if you can speak up now or not, but you mentioned that your question about race, I think, right, if I'm not mistaken, wasn't fully addressed. And if, well, we can go back to this and try to clarify it more. And if I'm not mistaken,
can I try to respond?
Yeah, it was a question to Dr. Kertai.
Yes, we worked on this analysis. Dr. Kertai, is it okay if I try to respond? Let me try. Look, this is the question about correlation between race and our outcome. Our outcome is Alzheimer disease risk. Of course, we know that if we use regression, our predictor, yeah, this is anesthesia risk, anesthesia level, and also add some covariates like race, we need to check if we have actually correlates or remaining correlates between race and outcome. Maybe we have, but we did also analysis in strata by races. So in this case, we have result independent of this correlation. Also, we try to compare the results from multivariable analysis when we use both anesthesia risk and races as covariates and many other covariates Miklas demonstrated. We also compared with results from univariable analysis when we use only anesthesia level as a predictor. And we found consistency between all this analysis. So I believe that possible correlation does not impact very our result. Also, Alexander asked some question about confidential intervals. I promised to respond. And he asked why we have so increased confidential interval from point to point. Actually, this is a data set with different time of follow-up, and for example, death or different causes of the end of follow-up are considered as independent censoring. And we have different statistical power for different points. And it's lower starting from the colorectal cancer onset. And that's why we have increased confidential intervals. And also we have some specific situation concerning delirium versus other predictors. Delirium is something specific. This is like acute event, and we consider it two different definition for delirium in the first year and delirium of the remaining years. This is approximate picture of why we have this level of confidential intervals and these dynamics.
Okay, thank you. Basically, this the number of people tend to decrease substantially.
Yes, that's true, because of limited follow-up for a group of patients.
Okay, yeah. Well, thank you.
Okay, thank you, Igor, for addressing those two questions. Okay, I got a note from organizers that if I understood it correctly, they can like turn on your microphone to let you speak, but you need first to raise your hand. Otherwise, I will not be able to do so. So if you want to speak up and just click this raise hand button in Zoom and like chat with organizers if you want, and then they will make you this possibility. Okay, so let me go back to our panelists. So are there any questions to each other, I would say?
I have a question to Don Royal. Is he around?
Yes, he is, I see him.
Don, I would be very interested to read details about your methodology you demonstrated. Did you publish it somewhere, or maybe you plan to publish in the near future?
Yeah, well, I've been publishing on this consistently since 2012. So I've probably published 30 or 40 papers on this so far. And then since 2015, independent groups are picking it up and starting to replicate this. So like I said, 4 different groups around the world have replicated this in their data sets.
I would be interested to read the most methodological paper. Maybe you can send us a reference for the paper with the most detailed explanation of methodology, including this latent methods and
a latent variable approach has a lot of advantages for kind of dirty data. So these structural models were developed in actually the social sciences to look at survey data or that sort of thing. And so it helps you get rid of measurement bias, like Dr. Whitson was talking about visual handicap interfering with cognitive assessment. Basically, you could construct a latent variable, which is what all the cognitive measures share, and they can't all share bias due to visual handicap, because some of them are verbally biased and some of them are visually biased. So the latent construct has shed the bias due to either visual or hearing handicap, because it does not impact every variable and therefore can't contribute to the shared variance across the entire battery. Another really interesting thing is that whenever you have a latent variable that is indicated by a wide range of measures, that actually becomes a measure of intelligence, not domain-specific cognitive function. So we're used to thinking of dementia as a problem that impacts memory loss or executive function, but intelligence is a different animal. It's the shared variance across a wide range of measures. That's been demonstrated since 1904. It's one of the foundations of psychometric assessment. And it's interesting to me that the only path to functional outcomes in cognitive batteries appears to be through intelligence and not through intelligence-adjusted domain-specific variation. And then I think that is going to have implications for dementia's specific biomarkers, because you need to propose a biomarker capable of influencing performance on every single cognitive measure available. And biomarkers that fail to accomplish that aren't really biomarkers of intelligence and therefore may have no functional salience. So we're used to thinking of what that starts to do for me is divide biomarkers into those that act globally on the brain versus locally. A local process like a head injury or a particular circuit in the brain cannot impact every single brain function and therefore doesn't contribute to intelligence and therefore may have no functional salience. And that's essentially what we're finding across dementias. Someone yesterday spoke of different dementing syndromes. Well, what I'm finding is saying there's only one dementia syndrome, which is disturbances in this D construct. If you adjust for that, no other cognitive change has a functional outcome, and therefore it's kind of like the icing on the cake. A group in Colorado constructed a D homologue in the NAC, the National Alzheimer's Coordinating Center dataset, which is every patient ever examined by any federally funded Alzheimer's center, aggregated into one dataset with 25,000 cases, all sorts of different dementia diagnoses. So the homologue's ability to distinguish all-cause dementia from non-demented cases, so normals and MCI cases, was 0.96. So it very accurately distinguished any kind of dementia from any kind of non-dementia, but it couldn't distinguish any two diseases. In other words, the cognitive changes that distinguish Lewy body from frontal lobe dementia from vascular, vascular from Alzheimer, doesn't have anything to do with D. It's orthogonal to it and is therefore not functionally salient. It's like the icing on your cake. The cake is D, and it cannot distinguish any two dementias, because if they had a difference in their D score, they wouldn't be dementias. In other words, D is capturing dementia's essential nature, which is a global disruption of brain integration. And what resonates with me in other people's talks is how the biomarkers or the risk factors for dementia may be limited to things that are global, that globally impact the brain. So for instance, hypoperfusion might globally impact the brain, but stroke may not. Strokes are inherently local, and inflammation might globally impact the brain. We were talking about a biological signature of age as somehow impinging the integration of functions. So I think the biology of integration may be different than the biology of localized pathologies. And localized pathologies may actually be less relevant to dementia than the biology of integration, however that works, through hormones, through metabolic changes that impact brain structures across the entire brain and not regionally. So this approach is going to emphasize global mediators and maybe de-emphasize local insults and pathologies, like network pathology, for example.
Thank you, Don. Thank you very much for your answer.
Thank you, Dr. Royal. And I see you also raised your hand, so you probably have some questions.
Oh, no, that was just that I thought they had instructed us to raise our hand if we wanted to talk.
Oh no, actually, I got a message from organizers. They say actually it's only for attendees, because otherwise they can speak, and we can. Okay. Are there any other questions from the panelists? If not yet, then okay, let me ask a couple of questions, which I have postponed. Okay, the first question then would be to Dr. Royal to continue this conversation about your approach to this latent construct variable. So, as we communicated actually by email, you think that I mean, we can do such kind of measures in, let's say, health and retirement study. And I believe you meant like some kind of homologue. It means like we have some subset of what we probably can do from the set of variables we have. So can you elaborate more on that? So what do you think would be the most appropriate measures to include into such kind of variable?
That's kind of what I was getting at. Because intelligence manifests in every observable brain function, that's one of its features, then it absolutely doesn't matter what cognitive measures you have available. You can construct Spearman's general intelligence factor, or D, which is a piece of that, out of any battery. And we've demonstrated that across, like I said, 17 different homologs with dozens of measures, but we've explored it in our published work all the way down to the item set of a single test. So essentially every item on the mini mental state exam, for example, is a test of cognitive performance. Maybe it has a lot of measurement error, maybe it has a lot of bias, maybe it's not a very good predictor, but what they all share is still general intelligence. And from the item set of a single test, you can improve its ability to predict dementia and functional status by turning that into a latent D homolog. And so we've shown that in a couple of papers. So basically the HRS clearly has enough functional status and cognitive assessment in it that you would be able to build a D homolog. I actually wrote a grant proposal that wasn't funded to harmonize all the different HRS sister studies on D, and basically then, regardless of what country the study was being done in or what translation the tests are being done in, you would be confident that a person with this D score would have the same degree of dementia severity in any other HRS sister study. So I'm confident you could build D in that. The problem with building a homolog de novo is whether you can validate it. So a large epidemiological study like the EPI studies, for example, might have cognitive measures you could build D out of, but they don't have any independent clinical assessments to be able to say who has dementia, who's normal, who has MCI. So what you might be forced to do is cherry pick measures in HRS that are available in a better characterized cohort, maybe not a population-based cohort, but a convenient sample, and then see if you can export the parameter weights for the construct from the well-characterized cohort that you can validate it in to the population-based cohort. And we've done that too. We exported a D homolog from TARC, which is well-characterized, into the Hispanic EPIs and back again. And so we could build it from nothing more really than clock drawing in the mini mental and a measure of caregiver rated IADL, show that accurately diagnoses dementia in TARC, and then export the factor weights into EPIs. And suddenly you have an estimate of who is demented in EPIs, even though none of them ever saw a neurologist.
Okay, yeah, it's a good point, actually, yes.
Right. Another thing is we've published it somewhere that you could build it in a cohort in Texas and export the factor weights to Japanese data, the same measures, but obtained in Japan on Japanese persons in Japanese translation. And both of the cohorts were well characterized, and you could show that the ROC was identical in the two different cohorts. So exporting the construct from one well-characterized cohort to another one in a different country, in a different culture, in a different language. But the measurement bias inherent to the translation or cultural differences and how items are interpreted, all of that is measurement error that disappears from the latent construct because it doesn't impact all of the cognitive measures equally. It disproportionately impacts some measures more than others. And so that variance is in the residual of the latent variable. And so it's a convenient way to strip away measurement bias due to local factors.
Yeah, and yeah, I just want to comment. I wish your grant about harmonization of such kind of measures in HRS was funded, because it's really important to harmonize different data sets.
Yeah.
Yeah.
Well, I wish it was funded too.
Yeah. Okay. Thank you for a detailed response. Let me check again with our panelists or audience if there are any other questions at this point. Okay, if not, then I will continue asking questions. And this time I want to ask Professor Belsky a question about, I forgot what was the slide, but the slide which showed correlations between different measures which you had. And it's interesting that some of those are kind of like uncorrelated. It seems like they show a different aspect of this. So it's kind of like, they're not measuring the same thing basically, right? So yeah, that's that one, thank you. Yeah, like homeostatic like dysregulation, for example, is not correlated to Horvath's clock residual, for example, 0.05. So those are different things. So basically, I think it's kind of like it calls for like joint analysis, or maybe constructing some like overarching measure. I don't know how to put it, like to take into account different aspects of such kind of dysregulations, which are measured by different types of such kind of measures. So can you elaborate a little bit on this?
Well, I think, yes, I'd be happy to. I should say, you've just positioned me in between your audience and their lunch break. So I'll try to be brief. I admire your fortitude in pushing through to the end. We reported, I think our preprint came out in 2017. The paper was ultimately published in 2018. For the first time, this low correlation between physiology-based measures of biological aging and these first-generation epigenetic clocks, and also telomere length for that matter, which I haven't bothered to include here. And this has raised, some questions, among them those you put forward about whether this suggests we should be combining these multiple measurements to arrive at a fuller picture of biological aging. And I should say that there was recently an article published in eLife that we had a comment on that took this approach. They modeled 5 different ohms. They had metabolomic data, proteomic data, gene expression data, DNA methylation data, and telomere length. And they made models of chronological age with each of them. And then they found that when they combined those estimates, so they had a composite, which was computed as just the average of those 5 scores, it was more predictive, modestly more predictive of disease, disability, and mortality than were any of the ohm-specific measurements themselves. So one interpretation, there is that perhaps there is different information contained at these different levels of biological organization. I think in some sense that necessarily has to be true. There have to be some elements of biological aging not captured within different molecular dimensions that we might assay. But I think it's also possible that in the case of these first generation epigenetic clocks, the process they're measuring is fundamentally different from the process that's being measured in the other several measurements in this chart. So what causes DNA methylation states in cells to record the passage of time remains a mystery. It's a powerful and important biological mystery, but it's something we don't know much about. However, we do know that whatever causes that is only very loosely related to the process by which bodies deteriorate as they age, and certainly the process by which social determinants drive that deterioration to occur faster in some individuals than others. There are no observations of experimental manipulations of these first generation epigenetic clocks, although it's been observed that in cultured fibroblasts, they age many times faster than they do in regular living organisms. That's, there's a stark contrast with the second generation epigenetic clocks, which do show more correlation with these physiology-based assessments. And I think that ultimately it may be the case that rather than our needing to measure aging, with these poorly correlated measures put together, what we might be better served by doing is looking at where these measures intersect and how we can amplify that signal. And this, in some sense, is building on the same measurement theory that motivates Dr. Royal's model of D, which is that the signal may be common across multiple observations of a process. The noise will not be shared. And by focusing on the overlap across different kinds of measurements of the same construct, we'll refine our measurement and purge noise and get a better signal. So it could be the case that we should be doing is really, kind of adding the Horvath clock residuals onto homeostatic dysregulation. But I guess what I would say at this stage, particularly given the much weaker associations of the Horvath clock residuals with disease, disability, and mortality relative to the other measures, we might better turn our attention toward the second generation epigenetic clocks and some other measures if what we want to do is understand the process that is disabling and killing humans.